Okay, good afternoon, everyone. We are GreatBay-SCIE.
Did you know that molds are the hidden threat causing infectious diseases? Asthma, ABPA, hypersensitive pneumonitis, and even cognitive impairment, such as depression. All of these can be attributed to molds. Mold-related diseases affect millions. Spreading by airborne spores, indoor mold infestation reaches a prevalence of around 22% worldwide, with a formidable level of pathogenicity. Locally in Shenzhen, where the climate is high and humid, mold contamination accounts for 30% of all cases of childhood asthma. In the United States, the number of asthma cases attributed to mold is 4.6 million people, with a cost of $3.5 billion. Mold is everywhere. Where moisture exists, mold exists. With minimal nutrients, mold spores grow into mycelia, extending hyphae deep into the building wall surfaces, tissues, everything. Beyond a health issue, it damages building structure, destroys historical relics, causing to an annual loss of $40 billion globally. Current solutions include physical brushing, household chemicals, and oxidizers. Brushing removes only the mycelia, leaving the spores that can quickly grow back. Household chemicals such as soda and vinegar are also ineffective. Oxidizers jeopardize health and bleach surfaces, leaving people with increased risk of infection and extra expenses. It seems that molds are unstoppable, but in fact, it is not. Aiming to wipe out this pervasive public health problem from the source, GreatBay-SCIE 2025 developed a brand new mold eliminator, ArMOLDgeddon, targeting different structural components of the mold cells. Different from other fungicides, ArMOLDgeddon annihilates active mold growth in a safe and effective way.
Our engineering focused on developing a multi-component antifungal system targeting key structures of mold cell wall. Since chitin and glucan form a rigid protective barrier, we employ different chitinases and glucanases with human lysozyme and Geraniol to form a synergistic degradation and inhibition. Experiment follow the framework of expression and activity evaluation using SDS page and breath assay confirming protein U and DNS assay measuring enzyme activity. To target chitin, the structural component, 4 chitinases, RMV-CHI, BCCHA1, GLS-CHI-B, and PRCHI-A, were selected based on their antifungal ability and high chitinolytic activity. 3 yielded expression in E. coli with BCCHA1 and RMV-CHI possesses high specific activity toward colloidal chitin at 37 degrees Celsius. While our GLS-CHI-B did not possess any chitinolytic activity and PRCHI-A formed inclusion buddies. Therefore, we improved the performance through an AI model to redesign these 2 chitinases. Through the enzyme activity assay we developed, we found out that PRCHI-3 and 5 were identified as a soluble variant with measurable activity in the supernatant. For glucans, we employ multiple glucanases with synergistic specificities. BGLS27, BGLU1, and FLGLU30 each target different glycosidic bond in the mold cell walls. They were all successfully expressed in the supernatant and show catalysis ability. BGLU1 possesses the highest ability of 532 U per milligram and are able to react with most of the substrate within 10 minutes. Given that beta-1,3 glucan account for up to 80% of total glucan content, BGLS27 was also optimized for yield and activity. Furthermore, being inspired during an interview, we were also inspired that alpha-1,3 glucan also plays a crucial role in cell wall stability. Therefore, we expressed AGL-EK14. Unfortunately, we didn't obtain expected band and require further investigation. We also attempted expressing human lysozyme, which possess a antifungal effect through its cationic properties and oxidative mechanisms. Using P. pastoris as chases, protein expression is attempted via the genome integration to the His locus using a linear rice plasmid. Transformation is ongoing and future experiments will be underway. To enhance surface retention and utility, enzymes were fused with CVM2 module from Bacillus, increasing adherence to materials like wall paint and filters. The additional CBM not only clouded the PET and the cellulose slices strongly, which persisted after rinsing, but also increased the catalytic ability of both our chitinase and glucanase. Geraniol, a monoterpenoid, could disrupt fungal membranes. In the E. coli with PMVA plasmid constructed, we integrated the Geraniol synthesis module made up of AGG-PPS2 and an OBGS to allow the GPP producer directly convert it into Geraniol, boosting yield and efficiency. And additionally, by truncating the first 65 amino acid of the OBGS, we reached a 62.8% yield increase in the Geraniol. And reaction with the enzyme added with Geraniol was also confirmed that there's no negatively impact to the enzyme activity. To address Geraniol's volatility and hydrophobicity, we also produce gamma-CGTase to convert starch into gamma-cyclodextrin, forming more stable complexes with our Geraniol. Collectively, this module targets mold from different facets, forming a multi-layer antifungal system. Most of our enzymes were successfully expressed and verified. However, some of them the performance is still limited due to the structural constraint. Therefore, we employ modeling to guide rational redesign.
So with the aim of improving enzyme activity, soluble yield, and stability, we carried out optimization through a modeling approach, focusing on the catalytic domains of two kitinases, GLX-HIB, PRCHAA, and beta-1,3-glucanase, BGLS27. An innovative protein engineering pipeline is developed, which comprises 4 major steps: structure acquisition, design space selection, sequence redesign, and dry evaluation. As a central theme to our redesign pipeline, the third step, sequence redesign, is realized through deep learning-based modeling tools, protein MPN and ligand MPN. Protein MPN focuses on higher protein folding fidelity, while ligand MPN focuses on better interaction between the ligand molecule and the protein of interest. Okay. So the two kinases, GLX-HIB and PRCHAA, have solved protein crystal structures deposited, while BGLS27 has none and is thus modeled by AlphaFold to obtain the structure. After that, we carried out ligand docking and iterative multiple sequence alignment through HH splits to define active site and find the top 50% conserved residues. The rest non-conserved residues outside of the active site are set as input for MPN redesign. In total, 600 sequences are generated by MPN for all of our enzymes of interest. After that, we modeled these sequences using AlphaFold and assessed them based on a matrix containing PLDDT, Rosetta score, solvent accessible surface area, etc. These criteria ensure that variants with improved characteristics are favored. After evaluation, 6 best performing variants for each enzyme are selected as candidates for further wet lab validation. The wet lab results show that 5 out of the 6 PRCHA redesigned variants exhibit good soluble expression, with 2 showing chitinolytic activity. The highest yield is 262 mg per milliliter, sharply contrasting the inclusion body expression for multi-protein. This highlights the success in our modeling effort, and setting our redesign pipeline is a good starting point for future iGEM teams. In the future, we're going to investigate other tools and parameters to continue our modeling journey.
To make sure our project truly meets our need, we built our human practice journey around one goal: turning Armageddon from a lab concept into a responsible, socially considered solution. We engaged over 20 stakeholders across 5 progressive stages and 12 cycles, guided by the 4A principle: aspiration, action, analysis, and adaptation. We started by understanding the problem. Surveys across China show that 79% of people have battled household mold, frustrated by ineffective cleaning method and the pungent odor left behind mechanical product. Doctors and the Shenzhen CDC confirmed that mold causes serious infections and allergies. The feedback led us to focus on safety, long-term stability, and pleasant odor, choosing non-toxic enzymes and natural terpenes like Geraniol, and launching public education to raise awareness about mold hidden risks. Next, we identified our key users: hospitals, restaurants, and hospitals. Housekeepers and the restaurant owners told us that current methods are corrosive, smelly, and short-lived, while hospitals needed non-irritant, biodegradable solutions safe for vulnerable patients. We even tested existing products ourselves and experienced firsthand the eye and throat irritation they cause, confirming the need for a safer enzyme-based alternative. These insights guided us to design hardware prototypes like a lung spray hat for high ceilings and anti-mold AC filter for a humid environment. To ensure feasibility, we consulted experts across microbiology, engineering, and industrial design. They helped us enhance our enzyme through AI-based modeling, add alpha-glucanase to target mold spores, and ensure our approach stays safe and environmentally friendly. We also explored enzyme immobilization techniques and powder formulations to improve shelf life and simplify use, bringing Armageddon closer to real applications. To bring our solution closer to market responsibly, we interviewed investment experts and bioproduction specialists. We developed a sustainable roadmap: spray drying for stable powder product, high cell density cultivation for mass enzyme production, and microbial fuel cells to treat waste sustainably. This ensures Armageddon can be affordable, accessible, and eco-friendly. In our final stage, we expanded our impact. We reached out to cultural relics preservation experts to protect fragile heritage materials and the Maldives Hotel to adapt our design for a global humid climate. We also published the Armageddon booklet, an illustrated guide that teaches families and communities about how to understand and prevent mold safely, bridging science and daily life. Through each cycle of reflection, we listened, learned, and evolved. Our human practice transformed Armageddon from a project into not scientifically innovative, but also socially responsible, sustainable, and human-centered.
Through human practice stakeholders, the mold-affected patient confronted us with unexpected questions. Why use E. coli? Isn't it dangerous? What is synthetic biology? We realized her voice echoed a wider community, exposing a deep divide between cutting-edge science and public awareness. In order to bridge this gap, there is only one path: education. We set out not just to teach, but to connect, starting with the young. For primary students who think in shapes and stories, we turned genes into colorful puzzles and invited them to create their own microheroes. They didn't just learn about DNA. They imagined and created with it. Middle schoolers knew the terms, but not the real-world meaning. So we brought Symbio to their hand, extracting banana DNA, solving plasmid puzzles, and even visiting a real Symbio company. Over 95% left not just understanding synthetic biology, but feeling it. As learners grow, so do the challenges. Our high schoolers began with an offline lectures and an iGEM club, then delved deeper through newsletters and online classes to improve geographical and temporal diversities for public school lectures. To enrich the experience for international schools, we brought something new: Symbio Night, a lively GDCA and PCR singing session and ethical debates, and Symbio Orienteering, a hands-on collaboration games where participants team up to complete missions at each checkpoint, uncovering the principles of genetic bricks along the way. And for the broader public, we met in an everyday context. Through public talks, board games, and social media, we even invited parents into the lab to dye fabric using bacteria. Step by step, engineered organisms transformed in their minds from something abstract into something tangible, safe, and sustainable. Our educational approach grows like a living tree, rooted in 4 core principles: thorough, hands-on, interest-guided, and communication. These roots sustain everything we do. We move fluidly between theories, experimentation, and ethics, listening as much as teaching. The public has shaped conversation on real-world problems and future aspirations, while we've developed 3 types of teaching tools and 19 reading resources. These effects have taken root across 27 Chinese provinces. We don't simplify science. We bring people closer to it. Through trust and participation, synthetic biology becomes tangible. That's how we've built not just understanding, but a community imagining a future with us.
Mold isn't just a stain, it is a health threat. Where traditional cleaners rely on harsh chemicals, Armageddon wields the precision of synthetic biology. Our enzyme-based formula breaks down mold both gently and effectively, creating a safer living environment for everyone. But science is only strong if it serves a community. So we make conversations with doctors, families, and experts. We learn what safety truly means and continuously refine our project. We then took Armageddon beyond the lab. With experienced inventors and entrepreneurs, we gather realistic market feedback that validates our concept's commercial potential. While we developed the science, we didn't let spreading the seeds of understanding to the public. Armageddon is more than a product. It is a commitment to sustainable science and healthier futures.
Guys, one big round of applause, and thank you to the high school team from GreatBay-SCIE. Thank you guys so much.
Thank you, everyone. We are GreatBay-SCIE.
Great job. Wonderful job.